is Prabhakar, Associate Professor, Department of Pharmacology. Today we will discuss about the bioassay. So let us start with the definition. So comparative assessment of relative potency of a test compound to a standard compound on a living tissue. That means we are comparing a test compound with a standard compound on a living tissue, right? So qualitative identification and quantitative measurement of amount of active principle in pharmaceutical preparations or biological materials. So it is also the measurement of concentration of a drug from magnitude of its biological effect. Okay. So next, so what are the indications of bioassay, right? What are the significance of bioassay is the active principle is unknown. The active principle cannot be isolated to study the biological response of a new drug and to ensure the purity and potency, right? So if the chemical assay is not available or it is being a complex or insensitive to low doses, we have to perform bioassay to estimate the concentration of some endogenous mediators. So these are the indications where bioassay has to be implemented without any question. Okay. So first thing is the active principle should be unknown. The active principle cannot be isolated to study the biological response of a new drug to ensure the purity as well as potency. If the chemical assay is not available or complex and it is very insensitive to the low doses, and to estimate the concentration of endogenous mediators. So for these purposes, we have to do bioassay. Next, we have the principles here. So we have to compare the potency of unknown substance with the standard that we have been discussing throughout this bioassay. And we have a standard and test sample should have same pharmacological effect and mode of action. So we claim that our molecule is equivalent or with equal pharmacological effects and similar kind of mechanism of action, right? So these are the basic principles, and we have another set of principles like the tests and standards should be compared using a specified pharmacological technique, and method selection selected should be sensitive and also reproducible and should have minimized errors. Before that, biological variations and methodology also applicable. Right. So these are the principles of bioassay. So we have the classification of bioassay here. We have two major categories. That is, one is graded bioassay, in which there are several types of bioassays out there. Matching type, bracketing type, interpolation, and multiple point bioassays are there. And in quantal, we have direct endpoint assay, LD50 determination, etc. Right. So these are the two major categories that we classify the bioassay. So major thing is matching, bracketing, interpolation, multiple point, which falls under the category of graded bioassay. And in quantal bioassay, we have a direct endpoint assay and also LD50 determination. So these are the two types of bioassay we follow. So what are the methodology of graded biopsy? So if you are doing a graded biopsy, this is what the ideal methodological or experimental setup is. First, you have to check the apparatus and probably you have to calibrate for its proper functioning. Next step is to prepare the physiological solution. That is physiological salt solution that is very ideal to the environment where the tissue or isolated organ can survive, and the arrangement of instruments and adjustment of water bath according to the temperature the tissue can survive. We have to balance the lever at the fourth step, and we have to select the tissues according to the need, right? So according to the need we have to select the tissues. Next, we have the surgical process and the collection of required tissue and the tissue attachment to the water bath, relaxation time given to the tissue, and prepare the standard drug solution, serial dilution, select the lowest possible measurable concentration, prepare the DRC of the standard drug, prepare DRC for test drug. So we have select an assay method 3 or 4 point assays, and finally we have calculations and statistical correlations. So these are all the methodology of graded bioassay. And we have an example here for the quantal biopsy, that is direct endpoint assay. Threshold response, threshold dose producing a required response measured on each animal. Example, biopsy of digitalis in cats, hyperglycemic convulsions in mice, threshold dose is equal to period of infusion into weight, right? Concentration of this equal to total drugs, sorry, threshold dose divided by concentration into CST. So this is the calculations we have to follow. And we have finally we have the advantages and disadvantages of the bioassay. So advantages is the chemical assay is too complex, so we can use bioassay as an alternate. If the difference between the results, if there is a difference between the results, bioassay given more importance. And we have a relatively, when we have a relatively new drug, the toxicity is unknown, so we can perform biosis. So these are the advantages. Next, we have the set of disadvantages here, and which consists of time consuming and requires much skill. Biological variations still exist in biosis. So these are all the advantages and disadvantages of biosis. So previously, no, we have before the 2018, we have a set of experiments that have been performed in lab itself. So once after the arrival of the softwares, no, the assays, bioassays cannot be performed for the UG students. So in PG pharmacology, we can perform this experiments bioassay in live condition. We can smoke the drug, and we can do the physiological salt solution preparation, followed by the serial dilution of the standard drug, and preparation of organ bath, calibration of levers, mounting the tissue to the organ bath, and you can perform the experiments. So this is what we used to do in nowadays. Nowadays it is not possible. Only PG syllabus is having the practical of using the animals, right? So we can do all these biopsies with the help of XCOM software. We can do like set of experiments like biopsy of digitalis and all basic experiments like DRC of acetylcholine, 5-cysteine, everything is available in the software, right? So thank you all.